Section: Introduction. In this section, we discuss the role of large language models, LLMs, in various fields, including their application in scientific research. While LLMs are useful, they often produce plausible but incorrect information, a phenomenon known as hallucination, which raises concerns about their reliability. Interestingly, some suggest that these hallucinations might actually enhance creativity, which is crucial in drug discovery. This process is complex, requiring extensive testing and evaluation of numerous potential drugs, making it both time-consuming and costly. We note that previous research has utilized machine learning and generative models to aid in drug discovery. Additionally, we recognize that textual descriptions in chemistry can help improve model performance. Since drug discovery demands both specific knowledge and creativity, hallucinations could potentially be advantageous in identifying new molecules with unique properties. We explore the potential of LLMs for generating drug-related information. Our approach involves using LLMs to create descriptions of molecules based on their SMILES strings and comparing these with descriptions from a specialized model, MOLT5. Our findings indicate that LLMs often produce inconsistent information, but these hallucinations might still offer useful insights for drug discovery. We propose that rather than trying to eliminate hallucinations, we should investigate how they can be leveraged. To test our hypothesis, we design experiments that modify the prompts given to LLMs. We generate textual descriptions of molecules and provide specific instructions to predict their functions, comparing results with those from MOLT5. In our study, we evaluate 7 instruction-tuned LLMs and find that hallucinations can indeed enhance performance in drug discovery tasks. For instance, LLAMA 3.1 to 8b shows significant improvement over baseline models. We also examine factors influencing hallucinations, such as model size and generation temperature, discovering that larger models benefit more from hallucinations. Additionally, we find that generating hallucinations in different languages, particularly Chinese, yields notable improvements. Finally, we conduct a case study to understand why hallucinated text can enhance LLM performance, revealing that these hallucinations often contain relevant suggestions that aid in predictions. In summary, our contributions include a systematic investigation of hallucinations in drug discovery, validation of their potential benefits, and an exploration of the factors influencing their effectiveness. Section Summary. In this section, we explore the potential benefits of hallucinations generated by large language models, LLMs, in drug discovery, proposing that these seemingly incorrect outputs can enhance model performance by providing valuable insights. Our experiments with various instruction-tuned LLMs demonstrate that hallucinations can lead to significant improvements in tasks related to predicting molecular functions, highlighting the importance of prompt design and model characteristics in leveraging these effects. Section: Related work. In this section, we discuss the role of large language models, LLMs, in drug discovery. Many researchers have acknowledged the potential of LLMs and generative AI in this field. Some have reviewed how LLMs contribute to different stages of drug discovery, while others have highlighted their advantages and applications in drug development. We note that LLMs have been evaluated for various tasks related to molecule prediction. Specifically, some researchers have developed multi-agent frameworks for AI-assisted drug discovery, while others have utilized models like GPT-4o to curate datasets for drug toxicity. Additionally, prompt-based methods have been used to create natural language explanations for cancer drug discovery, and the relationship between SMILES strings and natural language has been extensively studied. In the context of LLMs, these models primarily serve as tools to aid drug discovery. Building on previous research that focuses on the potential of LLMs in this area, we aim to evaluate their capabilities and explore how hallucinations, instances where the model generates incorrect or unrelated outputs, can be integrated into drug discovery. We also examine the concept of creativity in LLMs. While hallucinations are often seen as negative, some researchers view them as a form of creativity. They have proposed frameworks to enhance LLM creativity and analyzed its impact on creative industries. We recognize that both factual and faithfulness hallucinations can contribute to innovation, similar to how Alexander Fleming's accidental discovery of penicillin led to significant advancements. Our work is the first to investigate how hallucinations affect LLMs specifically in drug discovery. For our task formulation, we define drug discovery as a classification task aimed at predicting whether a molecule has a specific property relevant to a disease, such as inhibiting HIV replication. To align with LLM requirements, we frame this classification as a next token prediction problem. We use a prompt template that includes the molecule SMILES representation and its natural language description, along with task instructions. The LLM processes this input to generate a predicted label, constrained to yes or no for binary classification tasks. In our experiment design, we explore how hallucinations from different LLMs impact performance. We maintain a consistent prompt template while varying the description component. We establish baselines where the description is either empty or generated by another model. We then assess how hallucinations from various LLMs influence the predictions, allowing us to evaluate performance changes based on the generated hallucinated text. Section Summary. In this section, we review the integration of large language models, LLMs, in drug discovery, highlighting their roles in molecule prediction and dataset curation, while also addressing the dual nature of hallucinations as both a challenge and a potential source of creativity. We formulate the drug discovery task as a classification problem, utilizing a next token prediction approach to evaluate how hallucinations from different LLMs impact model performance. Section: Dataset. In this section, we select 5 datasets from the MoleculeNet benchmark that help us classify and infer the abilities of molecules based on their biophysical and physiological features. These datasets focus on determining whether a molecule has specific properties relevant to the human body. The HIV dataset includes molecules tested for their ability to inhibit HIV replication. The BBBP dataset provides binary labels indicating whether molecules can penetrate the blood-brain barrier. ClinTox categorizes drugs based on their failure in clinical trials due to toxicity. CIDR, or the Side Effect Resource, contains information on marketed drugs and their adverse reactions, organizing side effects into 27 categories. Tox21 assesses the toxicity of compounds against 12 different targets. For data splitting, we follow established strategies. We use scaffold splitting for the HIV and BBBP datasets, which organizes samples based on their two-dimensional structures, while we randomly split ClinTox, CIDR, and Tox21. We reserve 10% of each dataset for testing. In terms of label selection and evaluation, we utilize the original labels for HIV, BBBP, and ClinTox since these are binary classification tasks. For CIDR and Tox21, we choose the label with the most balanced distribution in the test set. For CIDR, we check if the molecule causes reproductive system and breast disorders, and for Tox21, we look for activity against matrix metalloproteinases. We evaluate model performance using the ROC-AUC metric. Regarding our model selection, we choose 7 popular instruction-tuned large language models, LLMs, from 3 categories: general open source, domain-specific, and OpenAI models. The general open source models include Llama 3-8b, Llama-3.1-8b, Mistral-8b, and Falcon 3 Mamba 7b, all with around 7 to 8 billion parameters. We use ChemLLM-7b as our domain-specific model for comparison. For OpenAI, we select GPT 3.5 Turbo and GPT 4o. We apply default hyperparameters across all models for generating hallucinations, setting the temperature to 0.6 and the maximum token limit to 256. In our main experiments, we aim to determine if adding hallucinations to prompts enhances LLM performance compared to using only the SMILES string or descriptive text, and which hallucinations yield the best results. Our findings show that, on average, all LLMs except Llama 3-8b perform better with hallucinations than with just the SMILES string. Similarly, All LLMs except GPT 3.5 perform better with hallucinations than with the gold reference. Notably, LLAMA 3.1 to 8b shows the most significant improvement with hallucinations from GPT 3.5, outperforming the Smiles baseline by 18.35% and the gold baseline by 13.79%. Falcon 3 Mamba 7b also exceeds the baselines by nearly 10%. These results suggest that hallucinations can enhance LLMs in drug discovery, regardless of the model category. We further explore which LLM-generated hallucinations provide the most benefits. Our analysis indicates that hallucinations from OpenAI models lead to the greatest improvements. Specifically, hallucinations from GPT-4o boost performance by an average of 4.07% and 4.54% compared to the Smiles and Molt-5 baselines, respectively. Hallucinations from GPT 3.5 also improve performance by 1.6% and 2.08%. In contrast, hallucinations from Falcon 3 Mamba and LLAMA 3.1 result in slight performance declines. Overall, while most LLMs can enhance performance in drug discovery through hallucinations, GPT-4o stands out with the most significant improvements. Section Summary. In this section, we select 5 datasets from the MoleculeNet benchmark to classify molecular properties related to human health, employing various splitting strategies and evaluation metrics. Our experiments reveal that incorporating hallucinations into prompts generally enhances the performance of LLMs in drug discovery, with hallucinations generated by OpenAI models, particularly GPT-4o, yielding the most significant improvements. Section: Empirical Analysis. In this section, we build on our main findings that hallucinations can enhance the performance of large language models, LLMs, in drug discovery. We conduct further experiments to explore factors that might affect LLM performance when using hallucinations, specifically looking at model size, the temperature for generating hallucinations, and the language used. For model size, we assess 4 instruction-tuned LLMs, 1 billion, 3 billion, 8 billion, and 70 billion parameters from the Llama 3 models. We compare their performance using hallucinations generated by the other models. We find that while all models perform better than the Smiles baseline, the 1 billion model does not surpass the Molt-5 baseline. Generally, as model size increases, performance improves from the 1 billion to the 8 billion models. The 70 billion model does not exceed the 8 billion model's performance but still outperforms the smaller models. Notably, all models outperform the Smiles baseline when hallucinations are included. Thus, we conclude that larger model sizes enhance the impact of hallucinations, with a plateau effect around the 8 billion size. Next, we examine the generation temperature, which affects the randomness of the model's output. Using the 8 billion model, we generate molecule descriptions at various temperatures: 0.1, 0.3, 0.5, 0.7, and 0.9. Our analysis shows that higher temperatures lead to lower factual consistency and higher hallucination scores. While there is a general trend of worse performance with higher hallucination scores, the model performs unexpectedly poorly at a temperature of 0.3 compared to 0.5 and 0.7. Overall, performance across temperatures is similar, ranging from 54.20% at 0.9 to 57.10% at 0.1. Without hallucinations, the 8 billion model scores lower than any temperature setting, reinforcing our finding that hallucinations improve LLM performance in drug discovery. In our case study, we analyze a text generated by GPT 3.5 describing a specific molecule. We use the 8 billion model for this analysis, as it performs best with the hallucinated text. The molecule, represented as a Smiles string, has a complex structure with various atoms and functional groups. The generated text contains hallucinations, such as the incorrect statement that the molecule is made of hydrogen, which is not present. Additionally, it includes subjective claims about potential applications in drug discovery. We examine the attention scores from the 8 billion model, which indicate how much focus the model places on different tokens. The scores show that the model pays significant attention to the hallucinated content, particularly the elements and structure of the molecule. The unrelated phrase about potential applications also receives notable attention. We hypothesize that this subjective information boosts the model's confidence, thereby improving its performance in drug discovery tasks.